Hi, I'm Monte McGregor. I'm with the Center for Mollusk Conservation, and I'm going to be talking today about propagation of Venusta concentrucensis, the cumberland bean, using in vitro culture. I would like to first start with some acknowledgments of our staff, our funding sources, most of the U.S. Fish and Wildlife Service, and our agency funds, and all the people that have helped us in this process. And that's a lot of our partners, and especially our families. A little bit about Kentucky. We have over 100 species of mussels and 41 genera. Several threatened and endangered species, and we're going to be talking about one of them, the cumberland bean, and it's in the Cumberland River system. The focus area for this study again is southeastern Kentucky, upper Cumberland River, a lot of endemic species of fish, rudder mussels, and fish, and a pretty good size watershed. Kentucky Department of Fish and Wildlife started the Center for Mollusk Conservation in 2002. It's located in Kentucky River system in central Kentucky. We have a lot of capacity for culturing freshwater mussels and holding mussels, and we also have 2 branch facilities at Minor Clark Hatchery and in Lake Cumberland. Just a little bit about the life cycle of the cumberland bean. The males and females have sexual dimorphism, and the larvae, once they're released onto the host fish, which is mostly darters, takes about 2 or 3 weeks, and then the juveniles fall off and start their life, and they can reproduce as early as 15 to 18 months. They can live about 15 to 20 years in normal years. They also have a lure, a mental lure that allows them to be a specialist on darters, and they attack their host fish, which gets near the lure, and then the larvae are discharged from the ovisacs. Well, mussels are propagated in a lot of different ways. You can add mussels to a confined area and allow the nature to do their own thing. You can infest fish, release the fish. You can infest the fish and then take the juveniles. But we're going to talk about today about in vitro culture, where we're actually taking the larvae, putting them in petri dishes in an incubator, and skipping the fish. The purpose of this study was to develop a method to transform the cumberland bean without the use of its host. And we're going to compare some basic formulas to different mixed formulas of different serum types. And we want to test it for this rare species, which cumberland bean is pretty imperiled in Kentucky. And we also want to use the formula to use this to culture juveniles for restoration and recovery. The in vitro culture is something that's not new. It started back in the early 1900s with Dr. Max Maple Ellis. He's really considered the father of in vitro culture. And he basically took infested fish, cut the gills out, put them in some solution, and then the juveniles would come off. He produced massive quantities using this method. But it wasn't really until about the 1980s, Dr. Robert Hudson was hired by the Tennessee Valley Authority to actually develop. He was a fish culturist, and he's really what I consider the modern pioneer for in vitro culture. He used basically similar components what Ellis had. He added some components to it, used CO2 incubators in petri dishes, and was able to transform several species in an incubator without the use of a host fish. Today, at the Center for Mollusk Conservation, we do a lot of different mussel research projects, but we started the vitro project in 2004, realized it was going to be really difficult for us to do it, so we partnered with the University of Louisville, their graduate program. We've got a PhD student, Christopher Owen, who over the next 10 years graduated, became postdoc, and was hired on here at the staff to enhance these formulas. And we basically created formula that worked for a lot of species, and over the years we've tested over 70-something species that work in this in vitro culture media. Vitro culture allows for mass production, has high transformation rates sometimes if you don't have infections, and it has a great potential. We do a lot of work with toxicity testing where we send animals off to people, and they use these in vitro cultured lab animals, so it gives us a standard kind of a mussel. It's got a lot of potential for recovery, but there's a lot of research still needed as well. In vitro culture is basically taking larvae from the females and putting them in petri dishes in an incubator and with the right physiological conditions and allow them to transform in the petri dish. One of the ways we also do is we want to test different kinds of serum types. We want to collect some native fish, try to get some large rough fish that we can get a lot of blood from, and we'll collect the blood, extract the larvae, and put them in the petri dishes, and then adjust the mixtures. When we're looking at different kinds of blood components, we usually use carp because it's a common species, but we also compare that with other species. Here's an example of some common things that you're looking for: the hemoglobin, the plasma proteins, glucose and lipids are pretty important as well. And the pH is very, very important to keep it very, very close to what the fish blood is on the fish, typically around 7.6. Just a quick comparison of plasma and serum. Basically, the plasma and serum is the clear portion of the blood. The plasma is the blood that contains the serum plus the clotting factors, and the serum is the blood that remains after the clotting factors are removed. We typically use serum because we allow it to clot and then we centrifuge it to remove that layer. How we do the fish blood collection: we use boat electrofishers. We take the fish, cover them up with a towel, and then take a syringe, a 50 ml syringe typically, sometimes a 20, and we'll able to use a heparin line inside the syringe to withdraw the blood from the fish, usually a heart puncture, and then we allow the serum to clot, transfer it into a larger container like a 500 ml container, and then we eventually centrifuge that and store it in a 50 ml tube or smaller, and then we sterile filter it to 0.2 microns and store it in small aliquots so we can thaw it and use it in our serum collection. Here's an example of the fish like we use. This is a carp. The blood's being drawn out of the carp using a syringe. Well, this test we're going to be testing different mixtures. We're going to test rabbit serum, carp, buffalo, and then mixtures of the two. Typically, we want to use less fish serum because it's more expensive or we can't get it. So we can purchase rabbit serum. So we want to make sure that we can make the most efficient use of our serum. We also add other mixtures to our culture media, basically M199 media with serum added to it in a 2-liter mixture. We add sugars and amino acids, lipids, MEM vitamins, non-essential and essential amino acids, and of course an antibiotic mixture of 3 different kinds of antibiotics. Well, some of the results that we got, we basically in our control, which is basically M199 media without any type of serum, we were able to get no transformation. That's not surprising. But when we added the rabbit serum to it, we didn't get as much we didn't get any transformation at all. Some of this is due to fungus and other contamination. When we increased the amount of rabbit serum that's in the mixture, we were able to get a little bit of transformation. And we also did the same when we tested buffalo serum. But we didn't get great transformation, all less than 20% or so. So we started mixing different quantities. We added buffalo, carp, rabbit all together, and eventually we got a mixture that worked. Typically we use rabbit 2 parts to 1 part of fish, so half a part carp, half a part buffalo mixed with 2 parts rabbit, and that mixture seemed to get us the highest transformation. We had some as high as 91%, some of our dishes transformed, and with as high as averages of 70-something%. This allows us to produce a lot of juveniles in a small amount of space with the right mixture. Some of the 0% transformations really came from a result of contamination, and we think that maybe carp and buffalo can be used independently, but the best results we've gotten so far is when we mix them together. So what happens, you put the larvae into the dish, and over time they transform from the original larval to the juvenile form. You see the foot, the adductor muscles, the gills, and all that are transformed in about a 2-week period under normal conditions. Here's the start on the left, finish on the right. You can see they're quite different anatomically, and we're always looking for these adductor muscles and the gills right here. And here's the foot. Here's what juveniles look like when they first come off out of the media for a little bit. And this is just a few minutes out. We dilute these by taking the petri dishes, adding about 25% of sterile water from the systems they're going into, allow them to set for about 15-20 minutes, and then diluting them the rest of the way. And that method seems to work fairly well to get them out of the sterile environment into the non-sterile environment, into the grow out chamber. Well, we also took some of these juveniles that we cultured, and this example where we started with about 6,500 juveniles, and we put them in these static trays with a couple gallons, 6 to 8 liters, and we added about a couple mils of algae into these daily, aeration, and then over time, little over a month, they were about a millimeter. 12 weeks, they're anywhere from 3 to 4 or 5 millimeters, and at 16 weeks they can vary as high as 6 or 8 millimeters, but on average they're about 5 millimeters. We also added additional juveniles to Lake Cumberland cages, which is another approach that we use to rear animals. Here's the grow out systems we use for the small 2-gallon trays. We fed a mixture of freshwater and marine algae, 2 parts freshwater and 1 part marine. We raised the freshwater algae, and we purchased the marine algae. Here's what the juveniles look like after a few months growing in the trays in sand. The cage culture has been very promising. We're able to take the juveniles and put in these floating cages. Each cage system includes a floating rack with 5 cages, and we put these in for a few months, and then at the end of this month, end of the period, usually around the 1st of November, we're able to take and rinse the juveniles from the sand into these screens and collect them. We had very good survival. Here's an example of mussels, about 500 cumberland beans that came out of 1 cage, and these are just 5, 6, 7 months old, and they're already 15 to 20 millimeters. So very healthy juveniles with high survival. Just to give you an example, in a couple years, we at Lake Cumberland, we're able to produce quite a few juveniles that we later used in mussel restoration, and very high survival. Here's an example of one of the ones that we released with the tag on it to show that this can be done using in vitro culture and other methods. So in just a summary, in vitro is a method we can use in culturing mussels. We can produce large numbers without a host. We just got to watch out for infections. The formulas are very specific, and the mixtures can be with commercial sera reduced cost, and fish serum is collected from the wild, and our formula that we have, rabbit and carp serum, seems to be the best.